Thank you. I see you conveniently aborted my last name. Very smart.
I was practicing today.
I'm one of the faculty here in the Department of Surgery. I serve as the director of the Aortic Institute here at Yale. My topic today is the development of a novel biomarker platform for this condition in aortic aneurysm disease. Up to 5 to 6 million Americans have aortic aneurysm disease. It's mostly asymptomatic. This makes the diagnosis of this disease and condition very difficult. Aneurysms complications are quite catastrophic. They often present without any warnings suddenly, and they entail aortic rupture or dissection, which is nearly catastrophic, resulting in sudden death in over 80% of the patients who have this complication. It's often referred to as a silent killer. Once detected, which is typically incidentally done, lifelong surveillance is recommended for this condition, and surgical intervention remains the only treatment option for these patients. Current guidelines for aneurysm surveillance and decisions about ongoing surveillance versus surgical intervention are mostly based on imaging modalities, which only provide macroscopic information. To this day, we have no biomarker or blood test to help guide the management of this condition.
See the blood moving there?
So, aortic aneurysm disease, I'm sorry, I wish I could mute this.
Yeah. My goodness gracious.
Sorry, this is from the operating room, I apologize. So, aortic aneurysm disease, I'll skip to the next step. So what we see with aortic aneurysm disease is that there's huge clinical gaps with this condition because current guidelines that are based on, maybe the source volume? Sorry about that. Thank you so much. I apologize. So there's a huge mismatch in what the guidelines recommend and what we see in the clinical practice. One of the major problems with this condition is even though guidelines recommend intervention for aneurysms at 5.5 centimeters in diameter, where the normal aorta is about 2.5 centimeters, most patients who present with aneurysm complications have aneurysms less than 5 centimeters, which leaves a huge clinical gap to fill. There's also no distinction for guidelines regarding the location of the aneurysm. And the biggest problem is imaging modalities are often lag behind compared to what we see with regards to progression of the disease. So what we find is that whereas imaging guidelines give us macroscopic information, we have no molecular window into the disease progression process at all. This leads to the critical need for why we need some kind of a blood test or biomarker to help monitor for these patients. Most biomarker platforms so far to treat this condition have been blood-based, of course, and they've all looked at many microparticles such as exosomes, free plasma proteins, and nucleic acids in the blood as biomarkers for aneurysm detection and surveillance. But almost all these tests have not achieved any clinical penetrance because if you take a blood sample, it represents my exosomes, nanoparticles, and proteins that are released by all cell types into the bloodstream. So this just leaves a huge dilution factor that we have to account for. So what we said in our lab, and what's unique about our lab, is that we take the nanoparticles from a plasma sample and we added an extra step where we enrich for the exosomes that are released by the aortic endothelial cells. And that enables us to enrich the signal and represents the signal that's coming from the aortic endothelium over 100-fold compared to a whole blood analysis. So if you look at the aortic aneurysm wall, or aortic wall, there's two main cell types that make up the aortic wall. That is endothelial cells that line the inside, and then the vascular smooth muscle cells that coat around the endothelial lumen. We've now developed methodologies and validated this in mouse models of aneurysm development, where we've shown that when we profile these endothelial specific exosomes in the blood, we're able to detect aortic aneurysm disease and track it. We've now extrapolated that to the clinical setting, so I'll share some of that data. So we've now been successfully enriching endothelial cells, endothelial exosomes from aneurysm patients and control patients, and we completed a cross-sectional study of 40 patients with aortic aneurysm disease and matched them to 40 age-matched control patients who don't have aneurysm disease but have other cardiovascular conditions. When we looked at markers, we found significant differences. VE cadherin is an endothelial-specific marker. When we looked at VE cadherin expression in the aortic endothelial exosomes of aneurysm subjects compared to their matched controls, we found that at the protein and mRNA level, VE cadherin as a cargo of the endothelial exosomes was significantly decreased in the aneurysm patients. Another markers that we tested were what are called matrix metalloproteinases. These are a class of proteins that are very important in maintaining the integrity of the blood vessel wall, the aortic wall. And what we found with these was that these are upregulated in aneurysm conditions. I apologize. These are upregulated in aneurysm conditions. So if you look at aortic aneurysm tissue, MMPs are upregulated and they break down the aortic wall, leading to aneurysm development. Based on this, we actually looked at MMP expression in the aortic endothelial exosomes, and we found a similar pattern. We noted that in these aneurysm patients, MMPs are upregulated in their exosomal output as well. So taken together, we think that we have a new molecular window into this condition that can help guide management of these patients. The Blavatnik Accelerator Award, the project we wish to complete is a larger patient cohort. We wish to complete the cross-sectional analysis based on these markers we've already identified. Our labs has now performed next-generation sequencing and identified new microRNA biomarkers that are significantly altered in aneurysm subjects. We wish to complete that. And we're now also starting to look at patients who come in with aortic emergencies to look at these marker differences. Thank you.
Thank you.